Good evening, Dr. B. John ആണ്. ഒരു 10:05 ആകുമ്പോൾ സ്റ്റാർട്ട് ചെയ്യാം. ആൾക്കാര് ജോയിൻ ചെയ്തോണ്ടിരിക്കും.
ഗുഡ് ഈവനിങ് സാർ, ഗുഡ് ഈവനിങ്. നമുക്ക് 7:05 ആവുമ്പോൾ സ്റ്റാർട്ട് ചെയ്യാം.
ഒരു 5 മിനിറ്റ് നമുക്കൊരു ബഫർ ടൈം കൊടുക്കാം.
ഓക്കെ ഓക്കെ സാർ.
അതൊക്കെ സ്റ്റാർട്ട് ചെയ്യാം കേട്ടോ. ബാക്കിയുള്ളവരെ ഇടയിൽ കയറിക്കോളും. ഇപ്പൊ കുറച്ച് സ്റ്റാർട്ട് ചെയ്തോളൂ, നീ സമയം വേസ്റ്റ് ആക്കണ്ട. ഓക്കെ റൈറ്റ്.
Okay. So we will continue our session from our last class. I hope that everything is going well for all of you. The entrance preparation, everything is going well. Okay. So without wasting much time, we will start. So we have some leftover topics from last class. Okay, so a little bit of leftover topics from last class. I need everyone to answer in the chat box, so we can be a little bit more interactive. And I hope that my voice is clear and you can see my screen also. Okay, so the first question is pet fish shop owner. Okay, pet fish shop owner presents with an ulcer and multiple nodules were seen along the lymphocutaneous route. Okay, so what is this? The image is shown here. So you can answer in the chat box. Try to answer in the chat box. There is no problem even if you get wrong. So try to answer in the chat box now. So what is the answer? Okay, see, what is happening here is that, see, this is a pet fish shop owner. Okay, pet fish shop owner. Okay, so a pet fish shop owner who is presenting with an ulcer. Okay, with an ulcer. So a pet fish shop owner means he is in contact with the fish. Okay, okay, so one of you have answered it correctly, Varunka Raju. So the answer here is actually Mycobacterium marinum. Okay, see, one second, I have a little bit of a problem with my writing device here. Okay, no, it's fine. Okay, sorry for that inconvenience caused. Okay, so see, we have a pet fish shop owner. So he has been in contact with his pet fish. Okay, so what is happening here is that this is actually fish tank granuloma. Okay, fish tank granuloma. Fish tank granuloma is also known as swimming pool granuloma. Swimming pool granuloma. See, this swimming pool granuloma or fish tank granuloma, it is caused by Mycobacterium marinum. Okay, Mycobacterium marinum, which is a non-tuberculous mycobacteria. Okay, so so that is the answer here. Now there are some other options here. See, this Erysipelotrix ruseopadiae. Okay, see, Erysipelotrix ruseopadiae, it causes a disease known as seal finger. Okay, seal finger, which is also known as whale finger. Okay, whale finger, it is nothing but the swelling of the finger. Okay, if you come in contact with certain fish, okay, by fish bite or something, you will get a swelling in your finger. Okay, that is erysipelas in your finger. It is a severely painful condition, so that is caused by Erysipelotrix ruseopadiae, that is seal finger or whale finger. Now, Mycobacterium ulcerans is another non-tuberculous mycobacteria that causes a disease known as Buruli ulcer. Buruli ulcer. Okay. Now, Sporothrix shengi is a fungus and it causes a similar lesion. Okay. See, this is a linear pattern of lesion. A similar lesion can be caused by Sporothrix shengi. Okay. So, so we have discussed about the swimming pool granuloma. There are some terms starting with the swimming pool, okay, or swimming. See, for example, swimmer's ear, okay, swimmer's ear. Swimmer's ear is actually the otitis media that is caused by Pseudomonas aeruginosa. Okay, so that is swimmer's ear. Okay, then there is another disease known as swimmer's itch. Okay, swimmer's itch. Swimmer's itch is actually caused by schistosoma. See, the cercaria larva of schistosoma penetrates through the skin, and at the site of penetration, there will be itching. So that is known as swimmer's itch. Then there is swimming pool keratitis. Okay, swimming pool keratitis, and that is caused by acanthamoeba. So these are some of the terms we have come in contact with this when discussing this question. Now we will move on to our second question. See, a 60-year-old army veteran from USA visiting Varkala, India, presented with a splenic abscess. He fought for the USA in Afghanistan and Vietnam. What is your diagnosis? So what is your diagnosis here? You can answer it here. Very good, very good. You are answering very good. See, this is melioidosis. See, the term that comes in the question is Vietnam. See, melioidosis is also known as Vietnamese time bomb. Okay, Vietnamese time bomb. So Vietnamese time bomb is a disease that is caused by Burkholderia pseudomallii. Burkholderia pseudomallii. Burkholderia pseudomallii. See, Burkholderia pseudomallii, the selective media that we use for Burkholderia pseudomallii is ash downs media. Ash downs media, and this will form pink-colored, wrinkled or corrugated colonies in this ash downs media. That is a selective medium for Burkholderia pseudomallii. Now, the treatment for this Burkholderia pseudomallii, we can treat with the carbapenem. Okay, so we can treat with the carbapenem. There is an intensive phase. Okay, carbapenem, or we can use ceftazidime. Okay, ceftazidime. The intensive phase is for 2 weeks. Intensive phase is for 2 weeks, and then we have a maintenance phase. Maintenance phase. In maintenance phase, we can give cotrimoxazole. Cotrimoxazole. So this is that is a treatment for melioidosis, which is caused by Burkholderia pseudomallii. Now, what is this Glander's disease and Farcy? Glander's disease and Farcy disease, both of these diseases are caused by Burkholderia mallii. Burkholderia mallii. Okay, so that is mainly disease of animals. Glander's disease is a disease of horses. Okay, so that is caused by Burkholderia mallii. Now, the Shanghai disease or Shanghai fever, it is caused by Pseudomonas. Pseudomonas. Okay, so with that we will move on to our next question. See, we have covered meningitis in the last class. So what is left over is actually urethritis. Okay, urethritis. Now, a 27-year-old male patient with a thick, purulent urethral discharge. Gram staining of the discharge is shown below. Which of the following is not true? Okay, so before going to our answer, what is this organism that is shown here? See, this is urethritis caused by gonococci. So this is gonococcal urethritis. So how we came to know that this is gonococcal urethritis? See, we have to look at the gram stain. We are given a gram stain. See, we have discussed about how to interpret the gram stain. Okay, so similarly, we will see what is happening here. See, this is actually a cell that is the neutrophil or a pus cell. So that is a neutrophil or a pus cell. So this is also a neutrophil and or a pus cell. You can see the multilobulated nuclei here. So this is the multilobulated nuclei of the neutrophil. Now inside the neutrophil, what do you see here? What do you see? What is the organism? What is the color of the organism that you see? It is pink in color. It is pink in color. So it is gram negative. It is pink in color, so it is gram negative. Now, what is the shape of the organism? The shape of the organism is spherical. So it is cocci. It is spherical. So it is spherical. So it is cocci. So this is gram negative cocci. Now, what is the arrangement of this organism? See, look carefully. What is the arrangement of this organism? See, it is arranged in pairs. See, it is arranged in pairs. So two cocci are there. So it is arranged in pairs. See here, see arranged in pairs. Two cocci. If you look carefully, you can see that they are arranged in pairs. So gram-negative cocci arranged in pairs. Now you can also see that there is a gap in between these cocci. See, these cocci are placed like this, and there is a small gap in between the cocci. If you look carefully, that is because these cocci are kidney shaped. Okay, kidney shaped. So gram-negative cocci that are arranged in pairs and are kidney shaped. This is the appearance of gonococci. See, gonococci is a favorite topic, one of the favorite topic for NEET PG. Okay, they have asked several questions from gonococci. So this is how you identify gonococci from gram staining. So we have came to the conclusion that this is kidney-shaped gram-negative cocci in pairs, that is gonococci. Now coming to the options, what is the answer here? See, cotton swabs are preferred as it contains more natural fatty acids. In fact, cotton swabs should not be used. Okay, cotton swabs should not be used for organisms like gonococci because they contain these fatty acids. These natural fatty acids are inhibitory. They are inhibitory. They inhibit the gonococci. So we will not get the gonococci in culture. Okay, so the first option is wrong. First of the options are correct. Now, chocolate agar with antibiotics is used for culture. That is correct. Chocolate agar with antibiotics, that is your Thayer-Martin medium. Okay, Thayer-Martin medium, which is a selective media for gonococci. Now, gram staining is more sensitive and specific in men than women. You may think that it is a wrong statement, but it is actually true because in men, gram staining is more sensitive and specific than in women. Why there is a gender difference here? Because in women, there are certain gram-negative cocci that are colonizing the vagina. Okay, so the vagina is sometimes colonized by some non-pathogenic diplococci, like non-pathogenic gonococci. So if they are present, we may read it as false positive. Okay, so that is why gram staining is more sensitive and specific in men than women. Now, both sexual partners should be treated with ceftriaxone. That is a drug of choice for gonococci. Okay. Now, some extra points for gonococci. This is an oxidase positive organism. Oxidase positive organism. See, in its family, there is another member there that is known as meningococci or Neisseria meningitidis that causes meningitis. Okay. So, both of these organisms are oxidase positive. Okay. Now, the gonococci or Neisseria gonorrhea is actually positive for glucose fermentation. Glucose fermentation is positive, but maltose fermentation is negative. Okay, but Neisseria meningitidis, it is positive for glucose and as well as maltose. Okay, so that is how we differentiate. If we need to differentiate these two organisms, that is how we differentiate. Okay, but it is actually the two different pathogens, the two different organisms cause. One causes meningitis, another causes genital tract infection or sexually transmitted disease. Okay, now we will move on to our next topic now. See, a 30-year-old man with multiple sexual partners presented with a painless ulcer in hand. There is painless axillary lymphadenopathy also. So what is the diagnosis here? Come on, you can answer in the chat box. Please answer in the chat box. What is the answer here? You can do this. You can answer this question. It is a simple question here. So he presented with a painless ulcer in the hand, and there is also painless axillary lymphadenopathy. Okay, we have answers coming. See, actually the answer here is syphilis. Okay, actually answer here is syphilis. Now, how we come to syphilis, I'll explain to you. See, genito-ulcerative diseases. We are going to discuss about genito-ulcerative diseases. There are 4 diseases here. So the first disease is syphilis. Syphilis. Okay, syphilis that is caused by Treponema pallidum. Treponema pallidum causes syphilis. Okay. Now the second genito-ulcerative disease is chancroid. Chancroid, and that is caused by Haemophilus ducrei. Haemophilus ducrei. Now the third one is actually lymphogranuloma venereum. Lymphogranuloma venereum that is caused by an organism known as chlamydia. Chlamydia. And then finally, there is another disease known as granuloma inguinale. Granuloma inguinale, and that is caused by an organism known as Klebsiella granulomatis. Klebsiella granulomatis. Klebsiella granulomatous. So these are the 4 genito-ulcerative diseases. Now, how do we differentiate between these two, these 4 genito-ulcerative diseases? How can we differentiate? See, we can differentiate by looking at the type of the ulcer and the lymph node that is produced. Okay, the type of the ulcer. So ulcer is actually painless. Okay, painless ulcer and painless lymph node, painless ulcer and painless lymphadenopathy. That is a typical feature of syphilis. Painless ulcer, painless lymphadenopathy. So that is a feature of syphilis. Now, for chancroid, there is painful. The ulcer is painful ulcer and painful lymph node. Both are painful. So painful ulcer and painful lymph node. So that is the typical specialty of chancroid. Both ulcer and lymph node are painful. For syphilis, both ulcer and lymph node are painless. Okay. Now coming to the type of ulcer in syphilis, syphilis is actually having hard ulcer. So it is known as hard indurated ulcer, which is also known as hard chancre. In chancroid, it is actually soft ulcer but painful. So painful ulcer and painful lymph node. Now coming to lymphogranuloma venereum, it is caused by chlamydia and it is actually having painless ulcer. The ulcer is painless, but the lymph node is painful. So painless ulcer, painful lymphadenopathy, that is lymphogranuloma venereum. Okay, lymphogranuloma venereum. Now coming to granuloma inguinal, see granuloma inguinal is also known as donovanosis. Okay, donovanosis. So donovanosis is caused by Klebsiella granulomatous, and we have a painless ulcer. Painless ulcer. The ulcer is painless. And here there are no lymph node. Lymph nodes are not present in granuloma inguinale. Lymphadenopathy is not there. Instead, there is pseudobubo. See, bubo means it is lymph node. Okay, so swollen painful lymph node is known as bubo. So pseudobubo means there is actually granuloma formation. So granuloma formation in the inguinal region. So it looks like a lymph node, but it is not actually lymph node. So it is pseudobubo, but it is painful. It is painful. It is not actually lymph node. It is a granuloma. Okay, so that is painful. Okay, now coming to syphilis, we have a lot of methods for detecting syphilis. We have VDRL, the most common method that we use. Then RPR, RPR is rapid plasma reagent. So we use cardiolipin antigen. Both of this we use cardiolipin antigen, an antigen that we extract from the beef heart. Okay, cardiolipin antigen. Okay, it binds with the syphilis antibody. So both of them detects the antibody as it binds to this cardiolipin antigen. Now, VDRL, we have to use a slide, and it is a slide flocculation test, and we have to use a microscope. In RPR also, we are using cardiolipin, but cardiolipin is bound to carbon particle in RPR, so that we can see it with our naked eye. So these are non-specific tests because we use another antigen for detecting the antibodies. These are non-specific tests. There are certain specific tests like a TPHA, Treponema pallidum hemagglutinin test, and as well as there is another test known as FTAABS. So these are all tests for detection of specific tests for detection of syphilis. Okay, now chancroid, how the organism looks, the Haemophilus ducrei, how it looks in like a school of fish pattern. Okay, school of fish pattern, which is also known as railroad pattern. Okay, and now in LGV there is lymphadenopathy. See, lymphadenopathy is there, and there is a gap between two lymph nodes, which is known as groove sign. See, groove sign is a specialty of lymphogranuloma venereum. Okay, lymphogranuloma venereum. Okay, so that is a whole picture of genito-ulcerative disease, and now you can easily answer depending on the whether the ulcer is painful or painless, and whether the lymph node is painful or painless, if lymph node is present or pseudobubo is present. Okay, so that is it about it. Now these are some of the pictures that we have here. See the first picture. This is actually the groove sign. See these are two lymph nodes here, and in between you can see a groove. See, this is the groove sign. So groove sign which is seen in lymphogranuloma venereum. So groove sign. Now these are actually intracellular organisms present inside the macrophage here. See, what is this? This is actually the Donovan bodies, intracellular bacteria, intracellular Klebsiella granulomatis that is present inside the macrophage. This is known as Donovan bodies, seen in Donovanosis or granuloma inguinale. Now, the last picture, see here, this is a railroad pattern or rail.
ഒന്നും കേൾക്കുന്നില്ല.
സ്ക്രീൻ ഷെയറും കാണുന്നില്ല, സൗണ്ടും മ്യൂട്ടായി ഉണ്ട്.
ഇപ്പൊ കേൾക്കാമോ? ഓക്കെ, എന്നെ ഒന്ന് കോ ഹോസ്റ്റ് ആക്കോ സ്ക്രീൻ ഷെയർ ചെയ്യാനായിട്ട്.
കോ ഹോസ്റ്റ് ആണല്ലോ.
ഓക്കെ. ഓക്കെ, ഇപ്പൊ കേൾക്കാമല്ലോ.
സൗണ്ട് ഓക്കെ ആണ്, പക്ഷെ സ്ക്രീൻ വരുന്നുള്ളൂ. യെസ്, ഇപ്പൊ കാണാം.
Okay, so we will move on to our first question in mycology. See, a 30-year-old medical salesman who has been recently back from West Bengal presented with the cavitary lesions in the lung with lymphadenopathy. His bronchoalveolar lavage direct microscopy shows intracellular yeast and smear from culture at room temperature are given. Okay, so can you try to answer this question? So what is happening here? See, he is coming from West Bengal. Okay, he is coming from West Bengal, and he is having cavitary lesions in the lung with lymphadenopathy. Normally, this cavitary lesion in the lung with the hilar lymphadenopathy, see, it is seen in tuberculosis. Okay, but here it is not tuberculosis. They are saying that the bronchoalveolar lavage direct microscopy shows intracellular yeast here. So this is intracellular yeast. Okay, this is the alveolar macrophage, and inside that you can see certain yeast. Okay, very minute yeast that is present inside the the macrophage. Okay, so that is the intracellular yeast, and also the smear from culture at room temperature is given. Okay, so this is the smear from culture at room temperature. So we have two different type of organisms at two different temperatures. So this is actually from bronchoalveolar lavage. So the temperature will be 37 degrees Celsius. That is our body temperature. So the organism is showing two different morphology. The fungus, it is a fungus, and it is showing two different morphology at two different temperatures, and that is known as dimorphic yeast. This is known as thermal dimorphism, and this is dimorphic fungus. Okay, dimorphic fungus, because it shows two different morphology at two different temperatures. So that is dimorphic fungus, and all dimorphic fungus causing systemic infection will cause cavitary lesions or pulmonary infiltrate with hilar lymphadenopathy and skin lesions. If you see that, then most probably it will be dimorphic fungus. So this is dimorphic fungus, and we have intracellular yeast that is described in the question. Now, what is seen here? What we are seeing here? See, the most important thing that we see is in the background. What is this? This is hyphae. This is hyphae. See the hyphae here. It is very important. You can see the other structures also, but the most important structure is hyphae. So it is having hyphae at 25 degrees Celsius and yeast at 37 degrees Celsius. So this is dimorphic fungus. And what else are we seeing here? See, what we are seeing here is macroconidia. See, conidia are the vegetative reproductive structures of the fungus. So we are having macroconidia. And what type of macroconidia is there? See, over the macroconidia, there are finger-like projections. It is a rough surface. Okay, macroconidia is having a rough surface. So finger-like projections over the macroconidia, and this is known as tuberculated macroconidia. Tuberculated macroconidia. See, intracellular minute yeast, tuberculated macroconidia, dimorphic fungus. These are features of histoplasmosis. So the answer here is histoplasmosis. Okay, so these are some of the pictures of histoplasmosis here. So we see intracellular yeast. Okay, so intracellular minute yeast are there. Again, tuberculated macroconidia here. See the tuberculated macroconidia. So this is seen at 37 degrees Celsius, and this will be seen at 25 degrees Celsius. So this is the mold form. This is the mold form. Okay, mold form, which is having the hyphae, and this is the yeast form. Okay, and here also you can see that the tuberculated macroconidia here, tuberculated macroconidia. See this also tuberculated macroconidia. See the hyphae in the background. That is very important. Why that is important, we will come to know in the later slides. So hyphae, see the hyphae here. See the hyphae here. So hyphae along with the tuberculated macroconidia, that is a feature of histoplasmosis. See, histoplasmosis is also known as Darling's disease. See, histoplasmosis is also known as Darling's disease because it is discovered by a scientist known as Darling. Okay, so the presentation will be pneumonia along with the hilar or mediastinal lymphadenopathy. Pulmonary infiltrates may be seen. The treatment is actually liposomal amphotericin B. Liposomal amphotericin B is the treatment. Okay, so liposomal amphotericin B is the treatment. So that is histoplasmosis. Now coming to our second question. A patient receiving chemotherapy develops pneumonia with the skin lesions. Microscopy is given below. Culture shows white colonies at room temperature, which on microscopy showed hyphae with the conidiospores. What is the diagnosis? So skin lesions and microscopy is given. Okay, so pneumonia with the skin lesion, microscopy is given. So microscopy is given from where? From the skin lesion. So this is skin biopsy. So what is the temperature of the skin? It will be 37 degrees Celsius. And then they are saying us that at room temperature, it showed hyphae with the conidiospore. Again, two different temperatures, two different type of morphology. This is also a dimorphic fungus. So two different temperatures, two different organisms are shown. Now look at the diagram here. Look at the picture that is given here. So what is it? This is actually the yeast form. So this is yeast. Now, what type of yeast is this? This is budding yeast. You can see the mother cell and the daughter cell here. So this is the mother cell and the daughter cell. So this is budding yeast. But look at the type of budding here. This is known as broad-based budding. Broad-based budding. See the broad-based budding. See here. See the base is very broad. This is known as broad-based budding. Broad-based budding. So it will have a typical shape. Okay. So this is actually looks like a figure of 8. So figure of 8. It looks like a figure of 8. So an yeast that is with a broad budding, broad-based budding, and looks like a figure of 8 is actually blastomyces. Okay, so the answer here is blastomyces dermatidis. Okay, so we will come to blastomyces dermatidis. There are more pictures of blastomyces here. So blastomyces, it causes blastomycosis. See this look at this yeast. This is having that figure of 8 broad-based budding. They again figure of 8 broad-based budding is shown. See here, see figure of 8 broad-based budding. This is blastomyces, and now this is what is seen and when it is kept under 25 degrees Celsius, the hyphae with the conidiospore. But this is not important for you. This is not important. This picture will not be given. The picture that will be given to you is like this. Okay, the yeast form picture will be given to you. So this is blastomyces, broad-based budding yeast, figure of 8. This is also known as Gilchrist disease. Gilchrist disease or Chicago disease. Chicago disease. Okay, see all of these dimorphic systemic fungi, they are mostly originating from America, especially the desert parts of America. Okay, so most of them are named from that parts only. So this is also known as Chicago disease or Gilchrist disease. Okay, this will also cause pulmonary infiltrate, skin lesions are there. Now the detection, we can detect an antigen known as BAD1. Okay, so it is blastomyces dermatidis 1, BAD1 antigen can be detected. The treatment is liposomal amphotericin B, liposomal amphotericin B. Okay, so quickly we move on to our next one. A thermally dimorphic fungus shows barrel-shaped arthroconadia in its mold state. It causes desert rheumatism. So identify. So they have given in the question that it is a thermally dimorphic fungus and it shows barrel-shaped arthroconadia. Okay. So now what is this arthroconadia? See, this is actually the fungal hyphae. Okay. See, that is a fungal hyphae. And you can see that in between these cells, the cells in between got destroyed. Okay, so there is no cells in between. Okay, so only some parts of the hyphae are remaining. Okay, some parts of the hyphae are remaining. Such a hyphae is known as arthroconadia. Arthroconadia, which is also known as arthrospore. It can reproduce. Okay, see each one of these segments can reproduce or form into a separate fungus. Okay, so this is barrel-shaped arthroconadia. This barrel-shaped arthroconadia is a feature of Coccidioides immitis or coccidioidomycosis. Coccidioidomycosis, it is a very dangerous disease. So coccidioidomycosis. So this is actually arthroconadia. See, why is it called arthroconadia? See, arthroscopy means joint. Arthro meaning joint. So these conadia, they are actually joined together. These are all part of the hyphae, but at the junction or at the joint, they separate out and they form individual spores or arthroconadia. So this is a feature of coccidioides, coccidioidomycosis, and coccidioidomycosis is also known as desert rheumatism. Desert rheumatism. Again, dimorphic fungi, I told you, it originates from the desert parts of America. So these are some of the pictures of coccidioidomycosis. So it is a dimorphic fungi, coccidioidomycosis. It is caused by coccidioides. Okay, so this is the arthroconadia. See, this is seen at 25 degrees Celsius. Now at 37 degrees Celsius, we don't have yeast form for this fungi. Okay, like other dimorphic fungi, this do not have yeast form. Instead, they have spherules. Spherules. See, this is known as spherules. So in the body, if we take a sample from our body, we will see these spherules. Okay, so these are spherules. See, these are spherules. This is seen at 37 degrees Celsius, but 25 degrees Celsius, you see this arthroconadia. Okay, so arthrospore or arthroconadia, this barrel-shaped arthroconadia, this is a feature of coccidioidomycosis. Now, it is also known as desert rheumatism and also known as valley fever. Valley fever also known as California fever. California fever. So Valley, California. See again, desert parts of America. This also causes pulmonary infiltrates, skin nodules can be seen. The treatment is actually itraconazole. You can use amphotericin B also, but the mainstay is itraconazole. Now coming to the another one, a veteran scientist from Brazil was diagnosed with Lutz Splendor de Almeida disease. See, the name of the disease is funny. Lutz Splendor de Almeida disease. His tissue biopsy revealed the structure shown below. Positive organism. So what is the structure that we are having here? See, what is the structure? So the tissue biopsy is given. Tissue biopsy means the temperature is 37 degrees Celsius. And at 37 degrees Celsius, what do we see here? What do we see here? This is actually yeast. Okay, yeast. And what type of yeast is it? It is budding. This narrow-based budding yeast. See, narrow-based budding yeast. Okay, so it forms a structure like this. So have you seen a structure like this before? See, have you seen a structure like this before? See, this is actually a wheel. Okay, see, have you seen a wheel like this? A wheel like this. This is actually the wheel that you see in the boats and ships. Okay, so that wheel is known as pilot's wheel, or it is also known as mariner's wheel. Pilot's wheel or mariner's wheel appearance is actually a feature of paracoccidioides. Okay, paracoccidioides. See, these are the pictures. So this is what happens at 37 degrees Celsius. The yeast with several budding from its surface. See, I told you to look for the hyphae when we talked about histoplasmosis. It is because you may confuse histoplasmosis, tuberculated macroconidia of histoplasmosis with the paracoccidioides. Okay, so in tuberculated macroconidia, what you see, you will see the hyphae in the background, but here there are no hyphae. Okay, so there are no hyphae here. So this is actually the yeast form that is seen at 37 degrees Celsius of paracoccidioides. It causes paracoccidioidomycosis. Now this is what we get at 25 degrees Celsius or room temperature. This is the hyphal form, but not important. This will not be given to you. Only this will be given to you. Now there is another structure here. See, this is also again budding is narrow-based budding is. But what is happening here? See, narrow-based budding is. What is this? What is this character here? What is this character here? This is actually Mickey Mouse. Okay, so this is also another appearance. So Mickey Mouse appearance, pilot wheel appearance, mariner's wheel appearance, all these are features of paracoccidioidomycosis. So it causes a disease known as Lutz Splendor de Almeida disease, and it causes pulmonary infiltrate as well as skin lesions, and the treatment is itraconazole. Itraconazole is the treatment. Okay, so we move on to the next topic. So before we move on, we have now discussed systemic mycosis that are caused by dimorphic fungi. Okay, systemic mycosis causing dimorphic fungus. Okay, dimorphic fungi. So which are the dimorphic fungi we discussed? We have histoplasma. We discussed histoplasma. Then we discussed blastomycetes. Okay, blastomycetes. Then we discussed coccidioidomycosis and paracoccidioidomycosis. Okay, so these are the 4 dimorphic fungi causing systemic mycosis. Okay, now we move on. So we have to, apart from these 4, there are 2 other dimorphic fungi. Okay, so that we will discuss the 2 other dimorphic fungi. So one of them causes subcutaneous mycosis and one of them causes opportunistic mycosis. So coming to our question here, a young cat owner presented with the nodular lesions in his hand in a linear pattern. Histopathological examination and culture smear are given below. Identify the organism. So what is this organism? So young cat owner, nodular lesions. What is this organism? Now, see, there is a typical pattern that is happening here. See, this is a pattern. This is a linear pattern. So linear. So it spreads by a lymphocutaneous route. So it is given a young cat owner. So cat might have scratched its owner. Okay. So from traumatic inoculation. So trauma has occurred from the cat, and from the site we have a linear lesion spreading along the lymphocutaneous route. Okay. Now what is given here? Histopathological examination is given here. Histopathological examination means it is from the tissue. So the temperature is 37 degrees degrees Celsius. And what is seen here at 37 degrees Celsius? See, in the center, there is a yeast, and it is surrounded by eosinophilic radiating material. Eosinophilic radiating material. So it looks like the sun. So we call it as asteroid body. Asteroid body. Asteroid body. Okay, so asteroid body, and this phenomenon of formation of asteroid body is known as Splendor-Hipley phenomenon. Splendor-Hipley phenomenon. Okay, so the formation of asteroid body due to the eosinophilic material that is radiating around the yeast cell, that is known as asteroid body, and the phenomenon is known as Splendor-Hipley phenomenon. Okay, now at 37 degrees Celsius, see the yeast of this organism. See the yeast. See, it is elongated yeast, elongated cigar-shaped yeast. Okay, elongated cigar-shaped yeast. Okay, and this will be the picture of the organism at room temperature, that is 25 degrees Celsius. And what do we have here? What do we have here? We have a stalk and a flower. Okay, so see, it is a flower-like appearance, flower-like pattern, or a palm tree-like pattern. Okay, either pattern, whatever you see, you can say flower-like pattern or palm tree-like pattern. These are all features of Sporothrix schengi, or sporotrichosis. Now, deliberately cat owner has been given. See, this is also known as Ross-Gardner's disease. You might have easily answered if I have given Ross-Gardner getting this disease. Okay, with this linear pattern of lymphocutaneous root, that is Ross-Gardner's disease. But why I gave you cat owner is that nowadays the most common method of transmission of Sporothrix is becoming to be cats. Okay, see, we have cats as pets in our house, and these cats, the nails of the cat, we have Sporothrix colonizing the nails of the cat. So when you get a scratch from the cat, you can get sporotrichosis also. Okay, you can get cat scratch fever also, that we discussed earlier, caused by a bacteria known as Bartonella henselae. It will also have a similar presentation, lymphocutaneous spread, lymphadenopathy will be present. Okay, cat scratch disease. How we differentiate cat scratch disease from sporotrichosis? By microscopy and culture. In microscopy, we can see the asteroid body as well as the cigar-shaped yeast. Then at 25 degrees Celsius, you can see the hyphae with the flower-like pattern. Okay, so that is it. Now, Splendor-Hipley phenomenon is not a pathognomonic feature of our Sporothrix. Other fungi can also show this Splendor-Hipley phenomenon. So examples of other fungi are zygomycosis. Zygomycosis is our mucormycosis. Okay, mucormycosis. Then Candida can also show this Splendor-Hipley phenomenon. Aspergillus is another fungi that can show this Splendor-Hipley phenomenon. And then blastomycetes, we discussed blastomycetes, the blastomycosis, that can also show this Splendor-Hipley phenomenon. Okay, now we are moving on to our next question. A 30-year-old HIV patient presented with warty lesions in the face and trunk. Histopathological examination and culture at room temperature are given below. You have to identify. See, what is the lesion here? What is the lesion here? See the lesion. What type of lesion is it? See, it is umbilicated lesion. Umbilicated lesion. See, umbilicated lesion. So you may go for molluscum contagiosum here, because molluscum contagiosum is the most common organism causing this umbilicated lesion. But remember, it is not only molluscum contagiosum that can cause umbilicated lesions. Many other organisms can also cause umbilicated lesion. Okay. Now we have a histopathological examination. So histopathological examination means from the tissue. Okay. So this is at 37 degrees Celsius. 37 degrees Celsius, and the culture is given. This is not the microscopy, this is the gross appearance. Okay, this is the gross appearance. This is subrotes dextrose agar. Subrotes dextrose agar. So 37 degrees Celsius, what do you see here? These are yeast. And what type of yeast is there? Yeast with a central septum. See, yeast with a central septum, or a bisected yeast. This is bisected yeast, because most of the yeast actually divides by budding, but this organism divides by transverse fission. So bisected yeast. And what do we see in SDA, subroots dextrosagar? This is the fungal colony. Okay, see the hyphae, the fungal mold is there. A light greenish tint is also you can see, but around that you can see this red pigment. So red pigment producing fungus. All this points towards one organism, that is Talaromyces marnafi. Talaromyces marnafi. It was earlier known as Penicillium marnafi. The new name is Talaromyces marnafi, and it causes talaromycosis. It produces this type of lesions, skin lesions. It can also produce lung infections, pulmonary infections, automycosis, especially in immunocompromised patients. So red pigment producing fungus at room temperature and at 37 degrees Celsius, bisected yeast producing umbilicated lesion. It is Talaromyces marnafi. Okay, Talaromyces marnafi. Now what is here? What is here? This is Talaromyces marnafi at 25 degrees Celsius. This is the microscopy. That is the lactophenol cotton blue, LPCB. Lactophenol cotton blue is the stain that is used. So this is the microscopic appearance. And what do we see here? This is actually the paintbrush appearance. See, it looks like a paintbrush. This is paintbrush appearance, or also known as brush border appearance. And see, these are the conidia of this organism, that is the vegetative reproductive structure. These are not cocci. You may get confused as cocci, but see, this is lactophenol cotton blue staining, and you can see the hyphae here. See the hyphae and the brush border or paintbrush appearance that is typical of Talaromyces marnafi. This is also a dimorphic fungi. So these are the dimorphic fungi. First four of them we discussed. They causes systemic mycosis. Then Sporothrix shenghi, that causes subcutaneous mycosis, and Talaromyces marnafi, it produces opportunistic mycosis. And what is this? You see a little rascal sitting here chewing on bamboo. See, he is actually a bamboo rat. And bamboo rat is a reservoir for Talaromyces marnafi. So reservoir for Talaromyces marnafi is bamboo rat. But there is no evidence of spread from bamboo rat to humans. Humans get infection normally by traumatic inoculation or by inhalation. Now the treatment is amphotericin B. Okay, and I forgot one thing. The treatment of Sporothrix is actually itraconazole. So the treatment of Penicillium or Talaromyces marnafi is amphotericin B. Okay, so we move on to our next topic. A 68-year-old on chemotherapy developed dysphagia and chest pain. Esophagoscopy and gram staining images are given below. Which test is used to identify the species of this organism? So before going to the species, what is the organism here? So esophagoscopy is given. Okay, try to answer in the chat box. This is easy to answer for you. So here the esophagoscopy, and in esophagoscopy you can see white patches in the esophagoscopy, white patches. And see this organism here. What is this organism? These are yeast, gram positive budding yeast. See, mother cell, daughter cell, budding, mother cell, daughter cell, budding, mother cell, daughter cell, budding, gram positive budding yeast. And see certain certain yeast cells give rise to a hyphae like elongated structure. Okay, hyphae like elongated structure that is produced from the yeast. This is known as pseudohyphae. So pseudohyphae, but gram positive budding yeast cell with pseudohyphae. This is candida. Okay, so this is candida. And now how do we speciate candida? There are different species of candida are there. Candida albicans, then there are non-albicans candida like Candida krusei, Candida glabrata, Candida dublinensis. A lot of candida are there. How do we speciate candida? Okay, see the answer here is actually chrome agar. See, chrome agar, as a word implies, it shows different species of candida in different color. Chrome agar. So we can identify the species. If it is blue, we can say that it is one organism. If it is green, we can say that it is one organism. So that is chrome agar. Now BDG assay. What is this BDG assay? It is beta D glucan assay. Beta D glucan assay is a rapid diagnostic test for fungus. Okay, if BDG assay is positive, then we know that there is a fungal infection going on. we will know only a fungal infection. We don't even know which fungus. Okay, so that is beta D glucan assay, rapid diagnostic test for fungus. If BDG is positive, then that means that a fungal infection is going on. Okay, now there are certain exceptions for BDG. Certain funguses are negative for BDG. One of them is zygomycosis. Our mucormycosis is negative. Okay, mucormycosis is zygomycosis, or mucormycosis is negative for BDG assay. Now the second one is blastomyces. We have discussed the figure of 8 appearance, broad-based budding. Blastomyces, that is also negative for BDG assay. And cryptococcosis, Cryptococcus neoformans is a fungus, so that is also negative for BDG assay. All other fungus are positive for BDG assay. Okay, now galactomannan assay is a test, rapid test for Aspergillus species. Aspergillus, if galactomannan is positive, we know that Aspergillus infection is going on. Now GMS staining is a staining technique that we can use for fungus. Okay, it is only a staining technique that we can use for fungus. Okay, so GMS is gomori methenamine silver staining. Okay, gomori methenamine silver staining. So now see this picture here. See, this is actually the chrome agar. See, chrome agar. So see, different organisms in different colors. So see this is Candida albicans, and it is having a teal blue color. Okay, so teal blue color, that is a specialty of Candida albicans. Other organisms have different colors, so based on the color we can speciate. Now this is another method. In which we can speciate. This is known as germ tube test, which is also known as Raynaud broad phenomenon. So this is germ tube test. So what is germ tube test? See, germ tube test means we will inoculate the Candida species that we got in human serum, and we incubate for 2 hours. So human serum, 2 hours, what will happen is that if it is Candida albicans, it will germinate. See, this is continuous. Okay, so this is true germ tube. So this is germ tube positive, true germ tube. But look at this picture. We see there is a constriction. So this is pseudo germ tube. Okay, pseudo germ tube. There is a constriction here. So this is negative germ tube test. So positive germ tube test, there are 2 organisms, Candida albicans and Candida dublinensis. Okay, Candida albicans and Candida dublinensis, they are positive for germ tube test. All other Candida are negative for germ tube test. So we get an idea about the species. Now coming to the last picture here. See the bottom picture. See, this is actually Dalmo plate method. Dalmo plate method means we take the agar. The agar taken is corn meal agar. It is a nutrient deficient medium, corn meal agar. We inoculate the fungus and look the agar itself under the microscope. See, if it is Candida albicans, it produces this structure known as chlamydospores. This is known as chlamydospores, thick-walled chlamydospores. These are produced by Candida albicans. Several other organisms produce different type of morphology in corn meal agar. So Dalmo plate method can also differentiate the species of Candida. Okay, so that is the different methods for speciation of Candida. Now we move on to our next question. A 65-year-old priest residing in an old temple was admitted with the signs of meningitis. His CSF was stained and the microscopic image is given below. What is the stain that is used? So what is the stain used? Before that, we need to know what is the organism. So what is the organism here? See, what is there in the question? 65-year-old priest who is residing in an old temple. See, old temple, old church, old building. What are there in these old church, old buildings? What are there in these buildings? Okay, what are there? There are pigeons. Okay, so pigeons are there. And when pigeons are there, who is there? Cryptococcus is there. See the question. The question leading to the answer here. It's a beautiful, isn't it? See, the priest that is residing in an old building, he gets meningitis. So this is a fungus that causes meningitis, is known as Cryptococcus, that is associated with the pigeons. And Cryptococcus. See, Cryptococcus is a capsulated fungus. Cryptococcus is a capsulated fungus. And I have told you what is the best method for detection of capsule. We have discussed what is the best method for detection of capsule. It is latex agglutination test. What is the second best method for detection of capsule? It is negative staining. Negative staining. And what are the examples for negative staining? India ink staining. India ink staining. This is black colored dye. India ink. Then nigrosin. Nigrosin. Again, black colored dye. So these are negative staining. So this picture is actually negative staining. And what is there in this picture? See what is there in this picture. See the center. This is the organism. This is a round yeast. Okay. So round yeast. So round yeast of Cryptococcus, and you see a clear area, non-stained clear area around the yeast. This is the capsule. This is the capsule. So capsulated yeast causing meningitis. It is Cryptococcus. Cryptococcus neoformans. Okay, so that is answer. See, am I audible? Can you just write something in the chat box? Am I audible to you? Okay, good. So, so that is it. So it is a capsulated fungus. So Cryptococcus neoformans. So that is what we are having here. Okay, now Cryptococcus neoformans is actually BDG, beta D glucan negative, that we also discussed. And there are some special media for growing this organism, that is bird seed agar. See, it is associated with pigeon, so you can remember bird seed agar or Niger seed agar. See, this fungus actually produces melanin, so that can be seen in these agars, bird seed agar and Niger seed agar. Now the treatment is amphotericin B. So amphotericin B for CNS infections. Okay, for other infection we can give fluconazole, but for CNS infection we have amphotericin B. Okay, so now we move on to our next question. A truck driver presented with pneumonia. Lung biopsy histopathology examination is given below. So identify the organism. So what is there? What is there here? Can you answer this? What is there? See what is there. See, we are seeing hyphae, fungal hyphae are seen from the lung biopsy, fungal hyphae. See, can you see here? See, what type of hyphae are these? These are septate hyphae. Can you see the septations here? See, septation. So septate hyphae. See the septations there? So septate hyphae. So this is septate hyphae. Now what is, well, see the branching here. See the branching here. What type of branching is this? This is acute angled branching. See, acute angled branching or narrow angled branching. Acute angled branching or narrow angled branching. Okay, so narrow angled, the septate hyphae with the narrow angled branching. Now anything else? See, one branch comes and gives rise to two branches. One branch comes and gives rise to two branches. One branch comes and gives rise to two branches. This is dichotomous branching. Dichotomous branching. So septate hyphae with a narrow angled branching and dichotomous branching pattern is seen in Aspergillus. This is typical of Aspergillus. If you see this image, it is Aspergillus. Okay, so there are 3 species of Aspergillus. Aspergillus causes several diseases like allergic bronchopulmonary aspergillosis, aspergilloma, cutaneous aspergillosis. A lot of diseases are caused by aspergillosis. It can cause automycosis. Okay, see, these are the 3 Aspergillus species that you need to be knowing of. So the first one here, see, the first one here is actually Aspergillus flavus. Aspergillus flavus. The second one here is Aspergillus fumigatus. Aspergillus fumigatus. And the last one here is Aspergillus niger. Okay, these are the 3 important species. And how do you differentiate these species? See, it may not be required at your level to differentiate the species, but I will tell you just, see, this structure is known as vesicle. This structure is known as vesicle. And see, the conidia generating structures are known as phyalides. And you can see here that the phyalides cover 3/4 of the vesicle. Okay, 3/4 of the vesicle. Only 3/4 of the vesicle is covered by the conidia. So that is Aspergillus flavus. Now Aspergillus fumigators, you can see the vesicle, okay, less than half of the vesicle is covered by the conidia. Less than half is covered by the conidia. That is aspergillus fumigator. So it looks like fumes arising from the vesicle. Fumes. That is why it is known as fumigators. Now Niger, it is black in color. Niger is black in color. Also the vesicle is fully covered. See, vesicle is fully covered by the conidia. So that is aspergillus niger. Okay. So this aspergillus species we have discussed, BDG, beta D glucan is positive. That is one rapid test. Now another specific rapid test is galactomannan. Galactomannan is positive for aspergillus. All aspergillus are positive for galactomannan. Okay. So that is it about aspergillus. Now the treatment for invasive aspergillosis. Invasive aspergillosis is voriconazole. Voriconazole. Others you can give itraconazole. You can give. Okay. So now we move on to our next topic. A diabetic male patient who recently recovered from COVID-19 presents with facial pain and teeth pain. So what is the diagnosis? This you can answer. See, everybody has to answer this in chat box. Ignore that picture that is given to you. Answer this in the chat box. What is the answer here? Diabetic male post-COVID presenting with a fungal infection. What fungal infection? What fungal infection? Diabetic male, post-COVID. Very good, very good, very good. It is mucor. It is mucor. It is mucor. Now, from the question itself, we know that it is mucor. Now, how do we differentiate mucor? If only the picture is given, how do we know that this is mucor? See, mucor. Do you see any septae here? There are no septations here. There are no septations here. There are no septations. This is not septae. Don't mistake for septate. This is actually another hyphae that is going behind. So this is S-septate hyphae, S-septate hyphae, S-septate hyphae. See, no septate, S-septate hyphae. So it is known as S-septate or posiseptate. Okay. So sometimes one or two septate may be seen, but it is very difficult to see the septate. So this is S-septate hyphae, and it is very broad. See, it is broad hyphae, broad ribbon-like hyphae, broad hyphae. And look at the branching here. It is not narrow-angled branching. It is wide-angled branching. See, it is almost right-angled. It is wide-angled branching. So wide-angled branching, broad S-septate hyphae. It is the specialty of mucormycosis. So this is mucormycosis. Don't get fooled by this. Round structures. This is actually the horizontal cross section of the hyphae. Okay, some of this is actually a tissue biopsy. So horizontally, when we cut, some of them get cut horizontally. So that those are the structures. So this is a mucormycosis. Okay, mucormycosis. So mucormycosis, it is actually BDG negative, beta D glucan is negative. We discussed it causes rhinocerebral mucormycosis and pulmonary mucormycosis, and especially in diabetic patients post-COVID. Okay, now the treatment is surgery. Surgery is very important because this is an angio-invasive fungus. It can invade through your blood vessels. Angio-invasive fungus. So immediately you take the sample and send to the microbiology lab. Do the surgery. Don't wait for the result. If you wait for the result, the patient will not wait for you. So don't wait for the result. So that is mucormycosis. So the treatment is surgery. Then surgery plus you can give liposomal amphotericin B. Posaconazole also you can give posaconazole. So this is the mucormycosis. Okay. Now coming to our next organism. HIV patient presents with pneumonia. Bronchoalveolar lavage, KOH was negative. GMS staining is given below. The Gomori methylamine silver staining is given. So what is this organism? HIV patient, pneumonia. It's an opportunistic infection. Gomori methylamine silver staining. And what is seen in this Gomori methylamine silver staining? See, these are cyst. Okay, this is the only fungus with cyst form. Okay, cyst is seen, and see there is a particular shape for the cyst also. See, this is, it looks like a crushed or crumbled. Okay, crushed or crumbled cyst. This is actually looks like a tennis ball. I mean, a table tennis ball that has been crushed. So it is known as ping pong ball. What ping pong ball? Crushed ping pong ball appearance. Crushed ping pong ball appearance in GMS staining. This is pneumocystis girovecki. Pneumocystis girovecki is the new name. Earlier it was known as pneumocystis carini. So the pneumonia was known as PCP pneumonia. So this is pneumocystis. Girovic is a crushed ping pong ball appearance in Gomori methylamine silver staining. Cyst is there. So what is there is that in the alveoli, what is happening is that this organism goes and recruitment of plasma cell occurs. So plasma cell are recruited. So plasma cells are recruited. So plasma cells are recruited into the alveoli. So that will result in a frothy exudate. Frothy exudate inside the alveoli. The sputum will be frothy exudate in the alveoli. So because plasma cells are recruited, this is also known as plasma cell pneumonia. Plasma cell pneumonia. Okay, plasma cell pneumonia. Now, frothy exudate all over inside the lung. So when you take a chest X-ray, you will see ground glass opacity, ground glass opacity, ground glass opacity in the chest X-ray. So these are all features of pneumocystis, and the treatment is cotrimoxazole. Cotrimoxazole you can give as a treatment and also as a prophylaxis for HIV patients. Okay, so cotrimoxazole is the treatment and prophylaxis. So that is pneumocystis. Now coming to our next question, a carpenter presented with a verrucous lesion on his right knee. KOH smear is given below. What is the diagnosis? So KOH smear is given below. What is the diagnosis here? So KOH is a colorless stain. Okay, but first of all, remember that KOH is colorless. It is used for tissue lysis, lysing the tissue, especially skin tissue. It contains keratin. We have to lyse the keratin. So we use potassium hydroxide, and it is colorless. Okay, so these are not yeast. You may mistake this for yeast, but these are not yeast. But look at the color of this. Look at the color. What color is this? This is copper colored, and it looks like a coin. So it is known as copper penny body, which is also known as metallar body, which is also known as sclerotic body. This is a specialty of chromoblastomycosis. These are not yeast cells. This is actually the metallar body or copper penny body. Okay, so they divide in all planes. So that is why they looks like this. So it is copper penny body or metallar body, copper colored. So this is chromoblastomycosis. See the carpenter, and it is a verrucous lesion. Verrucous lesion means it is a cauliflower-like lesion, and it presents on the knee. See, carpenter usually does his work on the knee. Okay, on the knee. So knee is exposed, and this organism transmits through traumatic inoculation. So by wood splinters might injure the carpenter, and from there this organism causes the verrucous lesion, and that is chromoblastomycosis. Now coming to our next question. A young lady presented to the cosmetologist with a macular discoloration on her back. KOH examination of skin scraping is given below. What is the diagnosis? Again, KOH, potassium hydroxide smear of the skin scraping is given below. What is the diagnosis? Can you answer this one? Okay, can you answer? You might have seen this in your dermatology posting. Okay, hypopigmented macular lesion. Okay, so can you answer? So what is in KOH? We'll see what is there in KOH. See what is there. These are hyphae. You can see the hyphae. Lot of hyphae are there. Hyphae. Now what else is there? See, small yeast are there. Small yeast forming a ball-like appearance. Okay, ball-like appearance. So somebody has answered it correctly. Karim, very good. This is pityriasis versicolor. Okay, pityriasis versicolor. And what are we seeing here? These are meat ball. Some foodie has identified this and said that this looks like a meat ball, and this hyphae looks like a noodles, which is also known as spaghetti. So this is the spaghetti meatball appearance of pityriasis versicolor caused by a fungus known as Malassezia furfur. Okay, Malassezia furfur, Malassezia globosa, two species are there. So this is the hyphae and the meatball. Okay, spaghetti meatball appearance. So with that, we come to the end of our mycology. See, I have almost covered the high-yield topic. There are some topics that are left behind, but you have to go and read. Okay, so we move on to the next topic. So the question is, a migrant laborer from Bihar presented with fever and hypopigmented skin lesions. Microscopy of bone marrow is given What is the diagnosis? What is the diagnosis here? Now, what is this picture? What is this picture that is given here? What is there in the picture? See, this is actually a macrophage, and inside the macrophage, what is there? What is there? You are seeing a nucleus and a kinetoplast. Okay, a nucleus and a kinetoplast, a nucleus and a kinetoplast, a nucleus and a kinetoplast, a nucleus and a kinetoplast. Okay, nucleus and kinetoplast, these are speciality of a mastigote. And a mastigote form is the diagnostic form of leishmaniasis or Kala Asar. Okay, this is the also known as LD bodies, Leishman-Donovan bodies, LD bodies. Okay, so a nucleus that is associated with the kinetoplast, this is a mastigote, that is the diagnostic form. LD body, that is the diagnostic form of leishmaniasis. So we will discuss about leishmaniasis now. Now it is transmitted by this organism. What is this organism? This is sandfly. Sandfly. There are two sandflies there. They have two sandfly. One is phlebotomus, and lutsomea. Okay, lutsomea. So these are the two sandfly. So it is transmitted by the bite of this female sandfly. Okay, what are the features? What are the features? See here, this is hepatosplenomegaly. This is actually the liver, hepatomegaly. Okay, hepatomegaly, and this is actually splenomegaly, a huge spleen. Hepatosplenomegaly is seen in this leishmaniasis. This is Kala, also known as Kala Asar, and this is hypopigmented lesions that is also there in this leishmaniasis. Now, what are these two? These are nodular lesions of PKDL. Okay, post Kala Asar dermal leishmaniasis. Post Kala Asar dermal leishmaniasis, a late manifestation. So hyperpigmented nodular lesions. So these are all pictures of leishmaniasis. Now, how do we diagnose leishmaniasis? We can look for an antigen, okay, known as RK39. Okay, so RK39 antigen and antibody detection. RK39 is a antigen for leishmaniasis, and we can culture this organism in a media that is known as NNN medium. NNN, Novi McNeil Nicholson medium. That is the media that is used for leishmaniasis. We can also use Schneider's Drosophila medium. Schneider's Drosophila medium that can also be used. See, both these media, we can grow trypanosoma also. Both leishmania and trypanosoma can be grown in NNN medium and Schneider's Drosophila medium. The treatment is liposomal amphotericin B. Liposomal amphotericin B. Earlier we used to talk about pentavalent antimonials, okay, but now it is liposomal amphotericin B. We can also use miltefosine. Miltefosine can also be used. Now, coming to our next question. A young traveler who recently visited the Amazon forest developed fever, hepatosplenomegaly, and lymphadenopathy. He gives a history of insect falling into the eye. Insect falling into the eye. Which insect have fallen into his eye? Which insect? Okay, which insect? This is actually the redouit bug or triatomine bug or assassin bug. So that has fallen into the eye. So see here the eyelid is inflamed. Eyelid is inflamed at the site of entry of this parasite, and this is known as chagoma. Chagoma. At the site of entry of the parasite, there will be swelling, and that is known as chagoma. If the site of entry is eyelid, there will be unilateral swelling of eyelid, and this sign is known as Romana sign. Romana sign is seen in trypanosomiasis. Romana sign. So this is actually Chagas disease. Okay, so it is transmitted by redouit bug. So this is the redouit bug or assassin bug. Okay, so it causes Chagas disease. Okay, now there is another one. Okay, so this is caused by Trypanosoma cruzi. Now there is another one here. What is this? What is this organism? It is our CC fly. And the organism here is Trypanosoma brucei, and the disease is African sleeping sickness. Okay, now what is this? This is the diagnostic form of trypanosoma. This is known as trypomastigote. Trypomastigote is seen in peripheral blood smear. Peripheral blood smear. See, the amastigote form of leishmania is seen in bone marrow, splenic aspirate, etc. But trypomastigote is seen in peripheral blood smear. And what is this? How do we identify this? You see one flagella here goes undulating. This is actually undulating membrane. So with that we can identify this organism easily. Now this is actually Winterbottom sign. Winterbottom sign is seen in African sleeping sickness. So African sleeping sickness, we can see Winterbottom sign, posterior cervical lymphadenopathy. Okay, so that is Winterbottom sign. Okay. Okay, now we move on to our next topic here. A patient presented with fever, pallor, and hepatosplenomegaly. He tested positive for HRP2 antigen assay, positive organism. See, this is easy. You can answer this one. HRP2 antigen is positive. See, HRP2 antigen is specific for Plasmodium falciparum. It is an antigen detection test for malaria. Okay, so antigen detection test for malaria, HRP2 is positive only for Plasmodium falciparum. There is another antigen known as PLDH or pan-LDH. PLDH is positive for all malaria. Okay, all malaria-causing organisms, falciparum, malaria, ovale, vivax, knowlesi, all of them. PLDH is positive, but HRP2 is specific for Plasmodium falciparum. Also, another one is there that is known as PFLDH. Plasmodium falciparum LDH, that is also specific for Plasmodium falciparum. That is antigen detection assay. Now we come to our next question. A patient from USA presented with a fever, hepatosplenomegaly, and pallor. His peripheral smear is given below. Identify. See, answer this question. This is very easy question. This is glaring at you, at your face. This question. Answer this question. Please answer this question. Even if you get wrong, no problem. Please answer this question here. Okay, so this is actually what you are seeing here. See, you are seeing here is RBC. Okay, and what is there inside the RBC? See this structure. This is actually a cross. This is Maltese cross appearance. And this is a feature of babesiosis, Maltese cross appearance. That is seen in babesiosis. It is transmitted by a tick. We discussed in last class. Which tick? Hard tick. Which hard tick? Ixoid tick. Okay, so transmitted by ixoid tick. Okay, so that is babesia. Now we move on to our next question. A 40-year-old man presented with chronic abdominal pain. His USG liver is given below. How will you manage? So what is there in USG liver? What is there in USG liver? What are you seeing here? See, this is a cystic lesion. A cystic lesion is there in the USG. And inside that, you can see the cyst wall has collapsed. This is the wall of the cyst. And that has collapsed. Okay. Now, this is known as water lily sign. Water lily sign can be seen in X-ray, CT, MRI, and USG. Okay. So, in all the imaging methods, this can be seen. So this is water lily sign. This is a feature for hydatid disease. See, hydatid disease is caused by Echinococcus granulosus, which is also known as dog tapeworm. So how do we treat this? See, treatment depending on the USG grading. Okay, so treatment is depending on the USG grading. So USG grading means this. If you see this water lily sign, then that is CE3. Okay, we see this water lily sign, that is CE3. If you see it, then it is cystic echinosis 3. Okay, cystic echinococcus 3, that is CE3. The grade is CE3. If water lily sign is present, that is CE3. Now, if we see only a cystic lesion with no cyst wall, that we call it as CL. Cystic lesion with no cyst wall, then that is CL. Now, if we see a cyst wall is seen with internal echoes, that is CE1. Cyst wall with the internal echoes, that is CE1, and it looks like a snowflake. So this is also known as snowflake sign. Okay. Now there is cyst wall Systole is there, but internal septations are there. Internal septations are there. So it looks like a honeycomb. Honeycomb. This is CE2. This is CE2. CE2. Okay, now CE3 we discuss now CE3 and 4. CE3 and 4. I mean CE4 and CE5. Okay, CE3 is the water lily. CE4 and 5 they are actually inactive, and CE5 is calcified. Okay, inactive, calcified, and CE4 and 5 the management is observation only. Observation only. CE4 and 5. Okay, now CE2 is actually honeycomb. Now what is this pair here? Pair is actually puncture. Okay, puncture, aspiration, injection, and reaspiration. So what we do is that we puncture this under USC guidance. We puncture this with a syringe and needle, and we aspirate the content inside this. Okay, aspirate it, and then what we do, we inject a scolicidal agent. Scolicidal agent is injected in. After some time it is re-aspirated. So that is pair. So pair is if it is a CL or CE1, or even if it is a CE3 uncomplicated water lily, then we can use pair. But pair is contraindicated in honeycomb. CE2, it is contraindicated. Okay, so that is how about the treatment of hydroxide. Now medical management, we can give albendazole. Along with pair we can give albendazole. So albendazole can be given. So if it is honeycomb, you can do surgical excision. Also, if it is a very peripheral lesion, a very cutaneous lesion, then we can do surgical excision, because pair can rupture. Okay, pair can rupture, and that will lead to seeding. Okay, so now the definitive host for this organism is dog. Definitive host is dog. Intermediate host is sheep, and man is an accidental host. Now there is an old test that was used before, a skin test, that is known as Cassoni's test. Cassoni's skin test is used for hydatid disease, but it is not used now. Okay, not used now. Now we move on to our next question. A patient from Alappuzha presented with swelling in the left inguinal region. Peripheral blood smear revealed N-shaped larva with no nuclei at the tail tip. Okay, N-shaped larva with no nuclei at the tail tip. See, where is he coming from? He is coming from Alappuzha. Alappuzha is endemic for what? Alappuzha is endemic for lymphatic filariasis. So it is either Wuchereria bancrofti or Brugia malai. But it is said that there is no nuclei at the tail tip. No nuclei at the tail tip means it is Wuchereria bancrofti. Okay, so we will discuss it in detail. We will discuss it in detail. So filarial worms. We can discuss the filarial worms. Mainly 4 filarial worms are there. So which are the 4 filarial worms? We have Wuchereria bancrofti, and Brugia malai, and then 2 more are there. That is known as Onchocerca volvulus, and Lova. So these are the 4 filarial worms. When we see a filarial worm, we look for sheath. If it has a sheath, see, sheath is present for Wuchereria bancrofti, Brugia malai, sheath is present, and Lova, sheath is present, but for Onchocerca, sheath is absent. So Onchocerca is an unsheathed filarial worm. So this is we are talking about the larval filarial worms. So unsheathed filarial worm is Onchocerca volvulus, and then we look for tail tip nuclei. Tail tip nuclei are absent in Wuchereria bancrofti. Tail tip nuclei are absent in Wuchereria bancrofti. It is also absent. Tail tip nuclei is absent in Onchocerca also. But Brugia malai and the Lova, Brugia malai and Lova, tail nuclei are present. Okay, tail nuclei are present. Okay, now coming to the treatment. For Wuchereria bancrofti, we give DEC. Brugia malai, we give DEC. Lova, we give DEC. But Onchocerca DEC is contraindicated. We give ivermectin. Contraindicated because it can cause severe allergic reaction. So ivermectin is given for Onchocerca. See, Onchocerca causes a disease known as river blindness. And lova, it causes calabar swelling or fugitive swelling, calabar or fugitive swelling. So that is it about the filarial worms. So filarial worms are over. Now we move on to our next question. A 30-year-old bodybuilder presented with myalgia and swelling in right biceps. Biopsy image is shown below. So biopsy image of the right muscle is given. So this is a biceps muscle. So what is there in the muscle? See here, what is there? See, a spiral organism is here. A spiral worm is here. This is Trichinella spiralis. Trichinella spiralis. Okay, so it is transmitted by eating uncooked pork, and Trichinella spiralis can present with myalgia. Also it can present with myocarditis, and the treatment is actually albendazole. Albendazole is the treatment. Now we have a test known as Bachman's skin test. Bachman's skin test is used for Trichinella. Okay, so that is it about Trichinella. Now we move on to our next question. HIV patient presents with abdominal pain and diarrhea. His stool wet mount is given and it detected no eggs. Okay, so no eggs are seen in the stool wet mount. Only the larvae is seen. This is a larvae that we are saying. No eggs, only larvae. Opportunistic infection. HIV patient. All this points to only one organism. Strongyloides. Why there is no egg in stool? Why there is no egg in stool? Because this organism is ovoviviparous. Ovoviviparous means its egg it doesn't lay eggs. Its egg actually hatches out in when it comes through its birth canal. So when it is giving birth, the egg hatches in its birth canal, and the larvae is given out of its genital pore. So larvae comes out of the genital pore. The egg is there, but the egg immediately hatches out into larvae. So that is ovoviviparous. So no egg will be seen in the stool. Only this larvae will be seen. Now this larvae, you can confuse it with our ancylostoma and nectator. So these are hookworm larvae. You can get confused between these, but see, the identification is that there is a buccal space, which is a very short buccal space here. Short buccal space in strongyloides. Okay, but for our ancylostoma and nectator, see, the ancylostoma and nectator are blood-sucking organisms. They will have a long buccal space, and then only the esophagus will start. Okay, long buccal space, and then esophagus starts. But for our strongyloides, there is a short buccal space, and immediately the esophagus starts, and there is a double bulb esophagus. Okay, so double bulb esophagus you can see in hookworm also. So with the double bulb esophagus you cannot diagnose. So this is strongyloides. Now there is a term known as larva currents. It is caused by strongyloides. So have you heard of larva migrants? See, larva migrants means normally the life cycle of a nematode will see that it may penetrate the skin, and it may move to the lungs, or it may move to the intestine. Okay, so that is the life cycle of depending on the organism. But sometimes this organism that penetrates, it wanders around. It can't find the target organ. It wanders around in the skin or other organs. It wanders around. That is known as larva migrants. Now the strongyloids will exhibit this larva migrants, and the larva migrants of strongyloids is known as larva currents. Okay, simple. It is a larva migrants phenomenon shown by strongyloids, that is known as larva currents. Now strongyloids can cause auto-infection. Auto-infection means it is already passed as larvae in the stool, so it can infect the same person itself. So that is auto-infection. Strongyloids exhibits auto-infection. Okay, so and then one more property is there, that is parthenogenesis. Parthenogenesis means the strongyloid female doesn't require a male to reproduce. Okay, it can reproduce by itself. That is parthenogenesis. It doesn't need a male to reproduce. Okay, so that is about strongyloids. Now we move on to our next question. A very important topic here. An HIV patient presents with abdominal pain and chronic diarrhea. His stool microscopy is given below. Now, what is this organism? What is this stool microscopy given here? What is this given here? See, blue background. blue background, in the blue background, see pink or red organism in the blue background. What is this staining? This is acid-fast staining. So, okay, this is the acid-fast staining. This is modified acid-fast staining for stool that is being done, and we are seeing acid-fast organism here. Acid-fast organism, and this is an acid-fast parasite, and the acid-fast parasite is Cryptosporidium. So, Cryptosporidium oocyst in stool is actually acid-fast. Okay, so that is the thing about it is an opportunistic infection in HIV. So Cryptosporidium. There are two other organisms in here, so we'll discuss that. So we have Cryptosporidium. So Cryptosporidium parvum is the full name. Cryptosporidium. Then we have Cyclospora. And cystoisospora. This is also known as Coccidian parasites, and all these parasites are acid fast. All of them causes diarrhea. Okay, now how do we differentiate between these? See, all of them, these Cryptosporidium and Cyclospora are round, but cystoisospora is actually oval. So cystoisospora is oval. Now, which is the smaller one? The smallest one is Cryptosporidium. It is only 4 to 6 micrometer. Okay, the second largest one is Cyclospora, 8 to 10 micrometer, and Cystoisospora is 20 to 33 micrometer. Now, for easiness, we will compare it with the RBC. See, the RBC, the size of an RBC is roughly around 8 micrometers. So, Cyclospora will be the same size of RBC. Cryptosporidium will be smaller than RBC. Cystoisospora will be larger than RBC and oval in shape. Okay. Now, there are the sporozoite. Okay, sporozoites are present inside the oocyst. Okay, so this is the oocyst, and inside the oocyst, sporozoites are present. For Cryptosporidium, 4 sporozoites are present. 4 sporozoites are present inside the oocyst. Okay, now for Cyclospora, so the sporozoites are present inside a sac. Okay, so 2 each will be present. So total 4 is there, but they are present inside a sac. Now cystoisospora is an oval organism, and there is the 2 sacs are there, and 4 sporozoites each are there in these sacs. Okay, so that is how we can differentiate it another way. Now, autofluorescence is shown by Cyclospora. Autofluorescence means it can fluoresce by itself. Okay. Now, cystoisospora can also show autofluorescence, but Cryptosporidium don't show autofluorescence. Now, autoinfection, just as strongyloides is present in Cryptosporidium. See, auto-infection causing organism, we have Cryptosporidium, strongyloides, then we have Enterobius, our pinworm, it can cause auto-infection. Then we have Trichuris. Okay, so those are the organisms that can cause auto-infection. Now, the treatment for Cryptosporidium is nitazoxanide. So, nitazoxanide is a treatment. For Cyclospora and Cystoisospora, we can treat it with the cotrimoxazole. It is not clotrimazole, it is cotrimoxazole. Sulfomethylprim pyrimethamine. So, that is the cotrimoxazole. So, trimethylprim pyrimethamine combination is there. So, that is it. The cotrimoxazole. So we can give that for treatment. So with that, we finish that. Now we will see some of the eggs here. So just to sum up, we will see some of the eggs here, and we can we identify these eggs. Okay. So now what is this egg? The first one, what is this egg? You see an eggshell, and inside the eggshell, what do you see? A segmented embryo. See the segments. A segmented embryo inside the eggshell. A clear space is also seen. And it is non-pigmented also. This is actually hookworm. Both ancylostoma and Necator. Both of them have this egg, hookworm egg. Now, what is this second one? See, what is this egg? This is plano-convex egg. Okay, see, plano-convex egg with the larvae inside. You can see the larvae inside. Plano-convex egg with the larvae inside, and it is colorless. It is non-pigmented. This is Enterobius or pinworm eggs. Now, what is this here? This one here, see what is this? You can see that there is three pairs of hooklets inside, and there is polar thickening and polar filaments are there. Okay, so these are features of tapeworm. Which tapeworm? This is Hymenolepis, Hymenolepis nana. Okay, so Hymenolepis nana. This is also non-pigmented. See, these are non-pigmented eggs, means non-bile stained eggs. Okay, so non-bile stained egg, hookworm egg, Ancylostoma and Necator. Then Enrobius, Hymenolepis. These are non-bile stained eggs. These are colorless. There are a lot of mnemonics out there. Neha, lot of NEHA, Neha, lot of mnemonics are there. But mnemonics, you can search the internet and find out. There is no need for me to come here and tell you the mnemonic. Okay, so you can make your own mnemonic also. And now what else is there? See, all of this, what we are seeing here, this is actually Ascaris egg. Okay, this is the fertilized egg of Ascaris, and this is the unfertilized egg of Ascaris, and it is yellow in color. So it is bile stained egg. Okay, bile stained egg. Now what is this? This is a barrel shaped egg with mucus plug, barrel shaped egg with a mucus plug at its end. This is trichuris egg. Again, it is bile stained yellow in color. Okay, bile stained yellow in color. Now, what is this here? This is an oval egg. Okay, oval egg with a terminal spine. This is schistosoma hematobium, and it is non-bile stained. Why it is non-bile stained? Because it does not come through the feces, it comes through the urinary tract. It is an organism causing bladder carcinoma, schistosoma hematobium. Now see this egg. This is schistosoma mansoni. See, there is a terminal knob. Okay, a knob is there. Instead of a spine, there is a knob. So that is this is schistosoma japonicum. So in japonicum, the knob is there. Okay, so this is schistosoma mansoni. In schistosoma mansoni, there is a spine. It is not a terminal spine, but it is located towards the periphery. Okay, so that is schistosoma mansoni. So these are some of the eggs. Okay, so with that we finish the parasitology and we move on to the virology. So what is this? A 25-year-old IV drug abuser came with jaundice and elevated liver enzymes. HBsAg and anti-HBs are negative. Anti-HBc, IgG and HBV DNA PCR are positive. What is the answer here? Can you try to answer this one? Okay, if you don't know the answer, no problem. We will learn the answer now. So what is this? This is actually hepatitis B. Okay, caused by hepatitis B virus. It is a DNA virus, and how hepatitis B is transmitted. Hepatitis B is mostly transmitted by needle stick injury, okay, or IV drug abuses. 30% is the chance of transmission for hepatitis B. It can also be transmitted by blood transfusion and organ transplantation. Okay, so how do we diagnose hepatitis B? We can diagnose hepatitis B by looking at antigen and antibody. And we have three antigens. Okay, three antigens for hepatitis B. Which are the three antigens? We have the first antigen, that is known as HBsAg. Okay, so surface antigen HBsAg. Now the second antigen is HBcAg, that is the core antigen. Now we have HBeAg, that is the pre-core antigen. So these are the three antigens. So when antigen is there, antibody is there. So corresponding antibody is for HBsAg, we have anti-HBs antibody, and for HBc we have anti-HBc antibody, and for HBe we have anti-HBe antibody. Okay, is it clear? Okay, now what is here? What is happening here is that the HBcAg is not seen in blood. It is not secreted into the blood. It is seen only in the hepatocyte. It is not found in the serum. So it is not there in the serum, and we use serum for diagnosis. So HBcAg is not present in serum. So we have two antigen and three antibody. So these are the markers along with the DNA. DNA, we can do DNA PCR and find the hepatitis B DNA. So hepatitis B DNA. So these are the markers: two antigens, three antibody, and PCR for detecting the DNA. Okay, so now we are coming to our dreaded graph here. Our dreaded graph we see. I have feared this graph when I was in my UG time. Okay, so what is there in this graph? This is actually very simple graph here. See, the first one, what is there? The first one that is rising here. First one, what is this? This is HBsAg. That is the first antigen. First antigen to appear in blood. Okay, first antigen. That is a first marker to appear in blood. That is HBsAg. HBsAg. And what is this? The second marker is also an antigen. Second marker is also an antigen. That is HBeAg. Remember, HBC antigen is not seen in serum. So we cannot plot the HBcAg in the serum. Instead, what is there? Anti-HBc is there. See, this is anti-HBc. Okay, so this is anti-HBc. I'm sorry. Okay. So it came back luckily. So this is anti-HBc, and anti-HBc we have IgM. In the initial phase we have IgM, and later phase we have IgG. Okay, later phase we have IgG. Okay, see now, see this, this is hepatitis B S antigen HBsAg. It rises, and then what happens? It falls and goes below detection level. And when it goes below detection level, who rises? Anti-HBs. So this is anti-HBs. Anti-HBs. Okay, so anti-HBs. Once HBsAg disappears, anti-HBs rises. Similarly, once HBeAg disappears, who rises? When HBeAg disappears, anti-HBeAg. So this is a simple graph. Okay, now we will make it much more simple after discussing this. Okay, so after discussing this, it will become much more simple. So a question is given for you. How will you diagnose hepatitis B? So for diagnosing hepatitis B, the first marker you will look for is HBsAg. HBsAg. This is the first marker you will look. The antigen, HBs antigen. That is the first marker that we will look. And if we found that, if we find that HBsAg is negative, there is no HBsAg given in the question. HBsAg is negative. What will we look for? We will look for anti-HBs antibody. Anti-HBs antibody we will look for. Okay, so anti-HBs antibody. See, anti-HBs antibody can be positive, okay, or negative. Okay, depending on anti-HBs antibody, we are going to diagnose here. Okay, so when anti-HBsAg is positive, we look for anti-HBc. Okay, the other markers, and anti-HBe, and the viral DNA. So DNA, these things we look for. So HBsAg negative, anti-HBs antibody positive, we look for anti-HBc antibody. And if it is negative, anti-HBe is negative, DNA is negative, only anti-HBs is present. This is vaccination. Okay, so the patient has been previously vaccinated. That is the only thing. There is no infection going on. Okay, now coming to another scenario. Anti-HBs is positive. Okay, anti-HBs is positive. Anti-HBc is also positive. Anti-HBe is also positive. Okay, anti-HBe, anti-HBe is also positive, but viral DNA is negative. This is recovery. Okay, this is complete recovery, or this is also known as late convalescence. Okay, so HBsAg negative, anti-HBs positive, anti-HBc positive, HBe positive, but DNA negative, that is recovery or late convalescence. Okay, now coming to another scenario here. So anti-HBs is positive. See, anti-HBc can be positive, anti-HBe positive, but DNA is negative. Okay. Sorry, I just made a mistake here. Okay, see, anti-HBC is negative. Okay, so anti-HBS is negative, anti-HBS negative, anti-HBC positive, HBE positive, anti-HBE positive. Okay, viral DNA can be negative. Okay, so this is actually early recovery. Early recovery. Okay, so this is not much important. Early recovery is not much important. But the last one, what is happening in the last one? See, HBS is negative, anti-HBS is negative, anti-HBC is positive, and anti-HBE is also positive. Okay, so and the HPV DNA is also positive. See, HPV DNA is the most important. This can be positive or negative, but HPV DNA is positive. See, anti-HBS is negative, HBsAg is negative, but DNA is positive. This is actually escape mutant. Escape mutant. And what is escape mutant? Escape mutant means there is a mutation in the surface antigen. So our routine test cannot detect the HBsAg. Actually, HBsAg is present, but our routine ELISA or CAR test cannot detect the HBsAg. That is why HBsAg becomes negative, but HPV DNA will be positive. So this is escape mutant. Occult hepatitis B will also present like this, but just don't concern about that. So this is the what is happening when HBsAg is given negative in the question. Okay, when HBsAg is negative, look for anti-HBs. If anti-HBs is positive, look for anti-HBC, anti-HBE, and the DNA. All of them are negative. Only anti-HBs is positive. That is vaccination. Now anti-HBs is positive, anti-HBC is positive, anti-HBE is positive, and DNA is negative. That is late recovery or late convalescence. And if anti-HBs is negative and anti-HBC and HBE is positive with the DNA negative, that is early recovery. And then we have escape mutant. Escape mutant means anti-HBs is also negative, anti-HBs is also negative, and anti-HBC and HBE can be positive or negative, but the DNA is positive. So that is escape mutant. Okay, so now coming to another scenario, we have we will look for HBsAg. That is the first one we look for. Now HBsAg is positive. HBsAg is positive. What will we look for? What will we look for if HBsAg is positive? We look for symptoms and liver enzymes. Okay, symptoms and liver enzymes, whether that is given or not. Okay, so whether symptoms are there, whether liver enzymes are elevated. Okay, now if symptoms are present or liver enzymes are elevated, if symptoms are present or liver enzymes are elevated, then that means infection. That means infection. Hepatitis B infection is there. Okay, so now we need to know whether it is acute and or chronic. We need to know whether it is high infectivity or low infectivity. So for determining whether it is an acute infection or chronic infection, we have to look for the type. Look for the type. Okay, type of HB anti-HBC. Okay, so anti-HBC antibody. Okay, so anti-HBC antibody type we look for. So this is an antibody. It can be IgG or IgM. If it is IgM, that is acute. If it is IgG, that is chronic. So that is it. So acute and chronic. So type of anti-HBC we look to know whether it is acute or chronic. Now what we will look for, we need to know whether it is high infectivity or not. High infectivity or not. For high infectivity, we look for HBeAg. HBeAg. Now if HBeAg is present, that is high infectivity. And if HBeAg is negative, that is low infectivity. Low infectivity. And this is how we classify. So HBsAg is positive, symptoms are present, liver enzymes are elevated, infection. Now IgM HBC, anti-HBC, IgM anti-HBC, that is acute infection. Now IgG anti-HBC, that is chronic infection. Now IgM HBC, anti-HBC with HBeAg present, that is acute infection with a high infectivity. Now IgG anti-HBC with HBeAg negative, that is chronic infection with a low infectivity. If HBeAg is positive, that is chronic infection with a high infectivity. Okay, see, that is clear. That is how we diagnose. Okay, now there is one scenario remaining, that is if symptoms are negative or liver enzymes are normal or not elevated, that is carrier. That is carrier. HBsAg positive but no symptoms, Liver enzymes normal, that is carrier. Now, if it is carrier, we again look for HBeAg, HBeAg. And if HBeAg is positive, then that is super carrier, super carrier. Now, HBeAg negative, that is simple carrier, simple carrier. Okay, so that is it. Okay, so with this we can diagnose, depending on what data is given in the question, we can diagnose it. Okay, so HBsAg is positive, but symptoms are negative. Liver enzymes are not elevated. That is carrier. But in a carrier, if HBeAg is positive, that is super carrier. See, super carrier was an old term that we use. Now we call it as chronic immunotolerant hepatitis B infection. Okay, chronic immunotolerant hepatitis B infection. Okay, and simple carrier was old term. Now we call it as chronic inactive hepatitis B infection. Okay, so that is how we diagnose hepatitis B. Okay, now coming to the treatment of hepatitis B. See how hepatitis B can be treated. See, treatment of hepatitis B is actually tenofovir and telbudin. Now, what is a prophylaxis? What is the prophylaxis? We can give vaccine as well as immunoglobulin. Both are available. Now, what is the vaccine available? It is a recombinant vaccine. Recombinant vaccine, that is hepatitis B surface antigen, that is produced by a recombinant DNA technology from Baker's yeast. Okay, so that is it. Now, what is the schedule for adult? For adult, it is 0, 1, and 6 months. Okay, 3 doses. And for children, we can give a birth dose, and then we can give 6, 10, and 14 weeks. Okay, so that is the vaccination for hepatitis B. Okay, so with that, we come to our next question. A patient is diagnosed with the hepatitis C. What is the management here? What is the management for hepatitis B? See, ribavirin plus pegylated interferon alpha. This was earlier used, but now it is not used because of the side effects. So now it is not used, and it is not effective also in suppressing the virus. But the treatment now is sofosbuvir plus velpatasvir. What is the sofosbuvir, velpatasvir? These are direct acting antiviral agents. These are newly discovered direct acting antiviral agents. Now there are 3 categories of direct acting antiviral agents. The first category is NS3/4A and NS5A and NS5B. So NS3/4A is also known as protease inhibitors. NS5B is also known as polymerase inhibitors. Okay, and the thing that you have to note is that the NS3A or NS4A, the antiviral start with or end with previr. Example, grasso, previr, parita, previr. See, previr. Okay, then simiprevir is there. So then it is previr. It is NS3A or NS4A. Now NS5A, we have the ending is actually asvir. It ends with asvir. For example, it is daclatasavir. Then lepidasvir. Then velpatasvir. See, asvir. Now NS5B, it ends with buvir, BUVIR, buvir. Example, dasa buvir. Then sofus buvir. So these are the directly acting antiviral agents that can be used for the treatment of hepatitis C virus. Okay, so now we move on to our next topic. A 14-year-old girl from Nedumangad, Kerala, presented with altered sensorium, vomiting, seizures. So what is this? This is encephalitis. MRI shows, this is very important, MRI shows bilateral thalamus involvement and basal ganglia involvement. Basal ganglia involvement endemic to Nedumangad, southern India, southern Kerala. So this is Japanese encephalitis. Remember, in Japanese encephalitis, It is typical involvement in MRI brain is bilateral thalamus and basal ganglia. Bilateral thalamus and basal ganglia are involved in Japanese encephalitis. Now, how do we detect Japanese encephalitis? We can use ELISA, IgM ELISA to detect Japanese encephalitis. Okay. Now, there is a vaccine available for Japanese encephalitis, a live vaccine using the strain SA1414-2. That is the SA1414-2 is the live vaccine that is available for Japanese encephalitis. Now see here, this is HSV encephalitis. Okay, HSV encephalitis, temporal lobe is affected. So temporal lobe, if the MRI shows temporal lobe affection, temporal lobe is affected, then that is HSV encephalitis caused by HSV1. So HSV encephalitis caused by HSV1. Okay. Now how do we treat HSV1? We treat by acyclovir. Okay, and the diagnosis is by PCR, HSV PCR. Okay. And there is no vaccine that is available for HSV. And what is rabies? See here, the next one is rabies. See, in rabies, brain stem is affected. See, if we take MRI of a brain, MRI brain of a rabies patient, brain stem will be affected. Brain stem will be affected. Now, how do we diagnose rabies? We diagnose rabies by antigen detection. Okay, we can use fluorescent antigen detection. So how do we detect the antigen? From where? From nuchal skin biopsy. Earlier we used to do corneal impression smear. That is outdated. We use nuchal skin biopsy. Nuchal skin biopsy is the diagnosis of choice. And post-mortem diagnosis, we can take the brain and look for Negri body. That is a inclusion body. Okay, so that is the inclusion body. Okay, so that is it. Now, rabies, just a short word about the pre-exposure prophylaxis and post-exposure prophylaxis. See, post-exposure prophylaxis, we give vaccine as well as, depending on the category, we can give the immunoglobulin. Equine immunoglobulin is there, human immunoglobulin is there, human is better. So, the vaccine we are using Thai Red Cross regimen. It is an intradermal regimen, Thai Red Cross regimen. So, we give 0, 3, 7 and 28 days. Okay, 0, 3, 7 and 28, and we give the injection at two different sites. So 2 doses at each visit. So total 4 visits and 8 injections. So that is the Thai Red Cross regimen. The intramuscular regime that is slowly being outdated, that is the SN regime. Okay, so this is the Thai Red Cross regime. In SN regime, there is only there is another one more day is there. 21 is also there, but for Thai Red Cross, the 21 is not there. So it is 0, 3, 7 and 28. Only 4 visits are required. And pre-exposure prophylaxis, we can take pre-exposure prophylaxis, and it is the injection is taken on 0, 7 and 21 or 28. Either 21 or 28 you can take the injection, 0, 7, 21 or 28, and it is one site injection. Only one injection is required. Okay. Now, if the patient has been previously vaccinated, either post-exposure prophylaxis or pre-exposure prophylaxis, he comes again with the dog bite. We can give 2 doses. Okay, 0 and 3. Okay, and one site is enough. One site. Okay, so that is about the rabies vaccination. Now we are coming to toxoplasma. See here, toxoplasma is written here. So what is this toxoplasma? It is a parasite. Okay, toxoplasma is a parasite. And what do we see in brain? See, we see random calcifications in the brain. Okay, random calcifications in MRI brain or CT brain we can see. Then it can cause chorioretinitis. Okay, chorioretinitis, and the diagnostic form is actually the tachyzoid, spindle-shaped tachyzoid, and it can be detected by a dye known as Sabin-Feldman dye. Sabin-Feldman dye is used for toxoplasma, tachyzoid, for identifying the tachyzoid. Now moving on to the next question. See, I will take a little more time and we will finish off it today. Okay, no pending topic will be there. We will finish off. Just be patient for a little more time. A thalassemia patient receiving multiple transfusions develop aplastic crisis causative agent, aplastic crisis, causative agent. See here, the causative agent, aplastic crisis in a hemolytic anemia, whatever hemolytic anemia, whether it is thalassemia or sickle cell anemia, whatever hemolytic anemia, aplastic crisis is caused by parvovirus B19. This is another favorite topic for the examiners, parvovirus B19. Now, what are the specialties of parvovirus B19? What are the specialties? So, parvovirus B19 will show slapped cheek appearance, slapped cheek appearance, slapped cheek appearance. From when children are infected, slapped cheek, the both the cheeks will be red in color. Then it is also known as fifth disease. Fifth disease. Okay, the disease is also known as erythema. Erythema infectiosum. Erythema infectiosum. Okay. So it causes aplastic crisis in hemolytic anemic patients, sickle cell anemia or thalassemia. Now, in immunocompromised patients, it causes pure red cell aplasia. See, it affects the red cell. So it causes pure red cell aplasia. Okay, pure red cell aplasia. If it is a congenital infection, if the baby gets this infection as a congenital infection, then the fetus will get non-immune fetal hydrops. Okay, similar to the hydrops is caused by the Rh incompatibility. So this is non-immune fetal hydrops. Okay, that is caused by parvovirus B19. Now it can also cause a glove and Stokes syndrome. Glove and Stokes syndrome. That is a papular purpuric glove and Stokes syndrome. Okay. Now parvovirus B19 is the DNA virus. It is the smallest virus. Okay, smallest virus infecting humans. Okay, smallest virus infecting humans. So now what else are there in the options? So we have EBV here. So we will discuss a little bit about EBV. Epstein-Barr virus. So Epstein-Barr virus causes infectious mononucleosis. We discussed in the last class about Paul-Bunnell test. It is a heterophile agglutination test that will detect infectious mononucleosis, but this is almost outdated. We have monospore test, that is a rapid test for Epstein-Barr virus, or we can do ELISA also. Now, what are the Epstein-Barr virus is an oncogenic virus. It can cause cancer. What all cancer can it cause? It can cause Burkitt's lymphoma. It can cause nasopharyngeal carcinoma. It can cause Hodgkin's lymphoma. It can cause non-Hodgkin's lymphoma. So it is a very oncogenic virus. It can cause several cancer. Now there are oncogenes. What are the oncogenes for this virus? Oncogenes. The oncogenes for Epstein-Barr virus, the main oncogene is LMP1. See, LMP1. It is a latent membrane protein 1 producing gene. Okay, coding for latent membrane protein 1. So LMP1. Then there is EBNA 2. Okay, so these are the oncogenes. So just because we have discussed the oncogenes, we will discuss about oncogenic virus, means oncogenic organisms. Not only oncogenic viruses, we will discuss oncogenic organisms. See, oncogenic organisms can be virus. Okay, so oncogenic virus or parasite, oncogenic parasite, or bacteria, oncogenic bacteria, or fungi, which are oncogenic or cancer causing. Now, oncogenic viruses, the most important oncogenic viruses are human papilloma virus, then Merkel cell virus is there, which causes Merkel cell carcinoma. Then we have Epstein-Barr virus that we discussed. Then we have HHV8. This is the Kaposi sarcoma virus causing Kaposi sarcoma. Okay, Kaposi sarcoma. It also causes primary effusion lymphoma. It can also cause a disease known as Castleman disease. Then we have hepatitis B virus, hepatitis C virus, then HDV, hepatitis D virus. When it combines with the hepatitis B virus, all of them causes hepatocellular carcinoma. Okay, now we have HTLV1. Okay, now we have HTLV, human T cell lymphoma virus 1, HTLV1 causing T cell lymphoma. Then HIV, which causes the HIV causes HIV-associated malignancies. Okay, so these are the oncogenic viruses. Now, parasites, coming to oncogenic parasites, we have Schistosoma hematobium. Schistosoma hematobium causing Schistosoma hematobium that causes bladder cancer. Schistosoma japonicum causes colon cancer. Then we have Clonorchis sinensis, which is a fluke. Clonorchis sinensis, it can cause cholangiocarcinoma. It causes cholangiocarcinoma. Then we have another fluke that is known as Opisthorchis viverni. So Opisthorchis, another fluke. It can cause hepatocellular carcinoma. See, it is important. It can cause hepatocellular carcinoma as well as cholangiocarcinoma. Okay, cholangio as well as hepatocellular carcinoma. So that is a parasites. Now bacteria. Coming to bacteria, we have H. pylori. We all know the bacteria named H. pylori. It causes gastric cancer. Okay, gastric cancer. Stomach cancer. It can cause. Gastric maltoma it can cause. Then we have Bacteroides fragilis. Bacteroides fragilis can rarely cause colon cancer. Now fungus. We have one fungus known as Aspergillus flavus. We have seen this fungus. This produces a toxin known as aflatoxin, which is seen when it is in moist condition. When grains are stored in moist condition, this fungi will grow, especially groundnut. Aspergillus flavus will grow and it causes hepatocellular carcinoma. It is hepatotoxic. So that is oncogenic organisms. Okay, so with that we conclude. Now we have one more organism we have, that is human papilloma virus. Human papilloma virus, very important virus. It causes cervical carcinoma. See, 9 serotypes are there, and 9 serotypes causes cervical cancer. Now the least common organism or the low risk organism causing cervical cancer are HPV 6 and 11. They are low risk. Okay, low risk. Now high risk organisms, high risk serotypes are 16 and 18 causing cervical cancer. See, don't misunderstand that 6 and 11 cannot cause cervical cancer. 6 and 11 can also cause cervical cancer, but it is low risk. The high risk is 16 and 18. Okay, high risk is 16 and 18. Along with the 16 and 18, we have 31, 33, 45, 52, and 58. Okay, see, no need to remember. So this forms the 9 serotypes. All these forms the 9 serotypes causing cervical cancer. Okay, now this the 6 and 11 can commonly causes anogenital warts. Now, what are the proto-oncogenes? So oncogenes are actually E6 and E7. So E6 inhibits the P53 gene, and E7 inhibits the retinoblastoma gene. Okay, so those are the proto-oncogenes. Okay, now there is a vaccine that is available. Vaccine is a recombinant vaccine. The recombinant vaccine uses L1 antigen of human papilloma virus is used. L1 antigen, and the vaccines that are available. Are Cervarix. You know this Cervarix, Gardasil, and Gardasil 9. So, see, Cervarix contains 2 serotypes, the high-risk serotype 16 and 18. Gardasil contains 6, 11, 16 and 18, 4 of them. Okay, and the Gardasil 9 contains the all 9 serotypes. All 9 serotypes is there in L1 antigen of all 9 are there in Gardasil 9. Now there is a new vaccine that is available now, that is known as Cervavac. This is developed by Serum Institute of India. It is developed in India, and it is same as Gardasil only. Okay, it contains 6, 11, 16 and 18. Okay, Gardasil. Okay, so if you haven't taken the vaccine, HPV vaccine, go and take the HPV vaccine, because it is recommended for ages 9 to 26. If you are more than 26 years of age, you cannot take the vaccine. Okay, so it is recommended in between 9 to 26. If you take after 26, you might have already got infected with the HPV, so there will not be much protection with the HPV. So 9 and 26 is the recommended age group for taking. So if you haven't taken, go and take the vaccine. So should girls only take the vaccine, or boys also should take the vaccine? See, both girls and boys should take the vaccine in between this age group. Why girls and boys? You may think that all cervical cancer can occur only in girls. See, this human papilloma virus can also cause anal cancer, penile cancer. Okay, so boys also go and take. Another thing is that if a boy is having HPV colonization, and if the partner or the female is actually not vaccinated, she can get infection from the male partner. So boys and girls should take the vaccine. 9 to 26 is the age group. Okay, so because we have cytomegalovirus in one option, so we have this one. This is the owl eye inclusion body, owl eye inclusion body of cytomegalovirus. Owl eye inclusion body, see, looks like an owl. Owl eye inclusion body of cytomegalovirus. This is the slapped cheek appearance, seen in Erythema infectiosum parvovirus B19. Okay, now we are coming to the end. Our second last question, rotavirus vaccine. A 20-year-old girl was admitted with urinary tract infection. While taking history, you came to know that her mother died of cervical cancer. What is your advice? What is your advice? See, what is your advice here? What is your advice? See, your advice will be HPV vaccination, nothing else. Go and take HPV vaccination. She is 20-year-old. Now, between 9 and 20-year-old, go and take HPV vaccination. Okay. Now, coming to the next question. A 6-month-old child with poor immunization history presents with watery diarrhea. What investigation will you do to confirm this diagnosis? See, 6-month-old baby, poor immunization history. What is the organism here? This is a virus. Which virus? Rotavirus. This is rotavirus diarrhea, and the diagnosis of choice for rotavirus is stool antigen detection. You can do stool antigen detection. Okay, you can do stool PCR also, or you can do electron microscopy, but the easiest method is stool antigen detection. So antigen detection by ELISA, you can do stool antigen detection for rotavirus. Now this is the rotavirus. See, what is this? This is the electron microscope image. And how does this rotavirus look like? Wheel-like organism. It looks like wheel. So rotavirus looks like wheel. Now we have vaccine. 2 vaccines are there. Rotavac. Okay, 2 vaccines that you should know. Rotavac and Rotacil. Both of them developed in India. Rotavac and Rotacil. Rotavac developed by Bharat Biotech. And Rotacil developed by Serum Institute of India. Rotacil. Both are live attenuated vaccine. Both are oral vaccine. Okay, both are oral vaccine and given in 6, 10, 14 weeks. 6, 10, 14 weeks. Okay, so that is about Rota. Then Rotarix and all are there. There are foreign made vaccines, but Rotacil is currently available in Kerala. Okay. So this is a vaccine. There is a syringe, but it is not an injectable vaccine. This is for providing the oral dose. Okay. Now, what test will you do? This is our last question. What test will you do? Will you choose to diagnose HIV in a 2-month-old baby born to HIV positive mother? This is the last question. Can you answer this question? What test will you do for diagnosis of HIV in a 2-month-old baby? Okay, in a 2-month-old baby. Okay, see the answer. What is shown here? See what is shown here. This is actually dried blood spot. See, for this 2-month-old baby or neonate, what we do is that we do heel prick, and the drop of blood is made into a blot. Okay, a blot, and we blot into Wartman paper. Okay, so Wartman paper. So this is dried blood spot, and we do DNA PCR from this dried blood spot. HIV DNA PCR. See, you might think HIV is an RNA virus, which is a retrovirus. Where does this DNA come from? See, even though it is a retrovirus, it has an enzyme known as reverse transcriptase. Reverse transcriptase. So when it enters the cell, it will get converted. The RNA will get converted to DNA by the reverse transcriptase. So that DNA we are detecting. So that is, if DNA is detected, that means it is infection. Infection is there. HIV DNA PCR is used for diagnosis in HIV, diagnosis of HIV in babies less than 6 months of age, okay, born to HIV positive mother. Now, antibody detection by rapid test. Why antibody detection by rapid test? Why is it not used? It is a routine test that is being used. See, it is a baby 2-month-old. See, it takes at least 6 months for the antibody to develop. Okay, so antibody. Otherwise, what will happen? Maternal antibody will be present. Maternal antibody will be present till 18 months of age. So it will give false positive. Mother is already HIV infected, so maternal antibody will be present. So it will take at least 18 months for it to disappear. Okay, so that is why the rapid diagnostic test that we are detecting the antibody that we are not doing. Okay, so HIV DNA PCR from dried blood spot. Okay, so now we will discuss briefly about diagnosis of HIV. Diagnosis of HIV. So now the baby, if the baby is less than 18 months or 6 months, so let's take it as 6 months. Okay, if the baby is less than 6 months, we do dried blood spot and we do DNA PCR. DNA PCR. If the baby is more than 6 months but less than 18 months, okay, we do rapid diagnostic test. Rapid diagnostic test can be done. But the test, if the test becomes positive, then we have to do dried blood spot and confirm with the DNA PCR. Okay, so rapid diagnostic test, if it is positive for 6 month to 18-month-old baby, then we have to confirm with the DNA PCR. Now for adults who are more than 18-month-old, so we call it as adult. For adults, we can do NACO strategy according to NACO guidelines. Okay, NACO strategy. So there are NACO strategies. There are actually 4 strategies: strategy 1, strategy 2A, strategy 2B, and strategy 3. So these are the strategies that are available. Now strategy 1. So strategy 1 is for blood bank. We all the strategy we do rapid diagnostic test. Rapid diagnostic test. We have 3 rapid diagnostic tests. So these rapid diagnostic tests should be very sensitive and very specific also. The first test that we do should be very sensitive. Okay, then only we can pick up all the HIV infected patients. So in the first strategy, we do only 1 rapid diagnostic test and it is used for blood bank. Blood bank. Now 2A is for surveillance. Surveillance. Okay, surveillance, we can call it as unlinked anonymous testing. Okay, surveillance. For surveillance, we do 2 rapid diagnostic tests. Now for strategy 2B is for symptomatic or suspected HIV. Okay, symptomatic or suspected HIV diagnosis. So the patient is having symptoms of HIV, opportunistic infections are there. In those patients, we use this strategy 2B. Okay, here 3 rapid diagnostic tests are used. And for strategy 3 is for asymptomatic individuals. This is the test that we do for all other requirements. For example, a patient requires surgery. We look for viral markers, HIV, HPSAG, HCV. The HIV, we test this method. Okay, that is strategy 3. 3 rapid diagnostic tests are used. Okay. Now the treatment of HIV we have discussed in the previous class. It is tenofovir lamivudine dolutegravir. Tenofovir lamivudine dolutegravir. Dosage is 300 milligram and 50 milligram. Okay, TLD. Okay. Now if we have to diagnose HIV in window period. Now what is window period? Window period is the time taken. Okay, time taken after HIV infection and appearance of antibody. So this is window period is for antibody detection test. Okay, so that is window period, time taken for the antibody to appear. And during window period, if we want to diagnose HIV during window period, we have 2 tests. One is HIV RNA PCR, HIV RNA PCR and P24 antigen detection by ELISA. P24 antigen detection by ELISA. HIV DNA PCR is the best method. Okay, now coming to prophylaxis of HIV. Prophylaxis of HIV, see, we don't discuss much about prophylaxis other than needle stick injury prophylaxis, but we have a new drug that has come out, very important new drug, that is successfully completing its phase 3 trial. It is known as lenacapavir. Lenacapavir. See, yearly two injections will prevent HIV. Okay, so it is being tested in high risk group and it is very successful till now. It is in phase 3 trial and phase 3 trial will be over by soon. So 2 injections are given yearly and you will be protected from HIV by these 2 injections. Okay, so with that, I conclude the class. We conclude our class now. So I wish you all the best for your exam. So all the best. Okay, all the best for your exam. No need for any tensions. Okay, so still we can bring out a big change. Okay, still we have around 1.5 months. A big change can be brought out. Okay, so study hard. No need for tension. Study hard and go write the exam. Okay, so all the best. Now microbiology, always revise. At least you have to revise once weekly. See, I don't say that microbiology is a what you say kadal. Microbiology ഒരു കടലാണെന്നൊന്നും ഞാൻ പറയത്തില്ല. ഞാൻ പറയുന്നത് microbiology ഒരു കുളമാണെന്നാണ്. Literally microbiology കുളമാണ്. Okay, it is a pond. Okay, see, pond. Microbiology is a pond. You can swim from one end to another. You can swim to the depth. Okay, but the only problem is that this pond is made up of mercury. The moment you step out of the pond, you are dry. Okay, nothing retains. Okay, so that is it. With that, I conclude my class. So thank you for cooperating. So shall we end the class now? Okay, very good. So we are ending the class now. And one more thing, if you have any doubts, you can contact me. Okay, so I will show my email ID also. See, my email ID is drmicroteacher@gmail.com. Okay, so that is my email ID, drmicroteacher@gmail.com. If you have any doubt, you can mail me in this mail ID. I'm more than happy to clear your doubts. And one more thing, after the exam, please send your questions. Okay, so that is a resource that I have. Please send the questions to this mail ID so I can teach your juniors as well. Okay, so thank you. So with that, we end our class.